All right, today we're going to talk a little bit about angiosperm reproduction. So that's the reproduction that occurs in flowering plants with flowers as sort of the main concept. So before we start with angiosperm reproduction like on its own, let's do a quick comparison of it to gymnosperm reproduction so we know sort of what area we're in and what things we're going to be focusing on. So in terms of both of their reproduction, they both do a number of things that are very similar to each other. They are both seed plants. So they both use seeds to house the embryo. And that's really what's important. The seed is the embryo that's going to be the new plant. And so that's a key concept. Now, they also have moved away from water for the other portions of their reproductive system. So they use pollen as the sperm container, essentially. That's the best way to think about pollen, is that pollen is where the sperm end up, and so that's able to be used and moved from one place to another. They also typically then contain multiple things. So they're also both heterosporous, as in they have microspores and megaspores, which are very clearly male and female, and in fact are used as a major part of the reproductive system. So we'll see that as we go to the angiosperm reproduction, even though you've probably seen it from me in other things. All right, so what makes them different? Well, we had to backtrack to the gymnosperms to remember what their deal is. If you'll remember, their deal is that the gymnosperm means naked seed. And so in this case, in fact, seeds are unprotected outside of seed coat. So they each have a coat which has the stuff in the embryo inside of it, but they are otherwise generally unprotected. The other major thing about gymnosperm reproduction, so we're not focusing on the plant itself, because there's a couple differences between the plants, is that wind is the major pollinator in the case of gymnosperms. Gymnosperms are mainly pollinated by wind. So angiosperms, on the other hand, these being the true flowering plants, if these seeds are unprotected outside of seed coat, in this case, seeds are protected by, and what they're actually protected by is the ovary that forms around the egg before it is fertilized, becomes ripened and increased. And so we actually call it a ripened ovary, although you probably also think about it as a fruit. So that's our fruit, is our ripened ovary. Sometimes they're squishy, sometimes they're not. So I don't want to say that it's specifically a fleshy thing, because sometimes they're hard. Achenes are a hard version of a fruit, but they're still protected. And then, of course, angiosperms are pollinated by animals and insects. And in fact, most of the time, they co-evolved with these to work together for this process. So that's a big part of it, is that both the fruits and the pollination end up relating back to animals, whereas gymnosperms, they're just kind of on their own. They do their own thing. Wind takes everything where it's going to go, and it takes care of it. All right. So now let's nail down some of this actual flower stuff. So this is my horrific drawing of a basic flower, but it will allow us to do a pretty good job of covering all of the parts. All right, we'll start all the way at the very, very outside of a flower. The very outside of most flowers are what look a little bit like leaves, but really they're sort of the protective outside. So if you think of a flower bud before it has opened up, it's typically got green around the outside. Most of the time, those are what opens up into these little green sections that we call sepals. So this would be a sepal, and there's usually multiple of them. Like I said, they're usually green, but not always. If you have a whole cluster of sepals, you have a calyx, and this is outside the other things. And so we call this the outside whorl. So a whorl is how we sort of are going to name the sections of the flower, and there's 4 of them. So we started with the calyx. All right, the next whorl is the one that contains the petals, and the petals are typically colorful. This is how we interact with the pollinators. So we attract them by a size and shape of the petals. So they often have color. If not, they might be pollinated by things that don't care about color, but they do still typically have size and shape because they are what partially attracts and allows a place for pollinators to sit. Therefore, H is our petal. And if you have a cluster of petals, they are often called a corolla, which is something that we use actually to talk about a essentially ring of color, which makes sense for petals. So that's our second whorl as we move inside. All right, we're going to move one more whorl inside, and to get to that, we're going to be looking at A, F, and G, which happen to be the male parts of the flower. All right, so A is the whole male part of the flower, and we give that the name stamen. And there's usually a bunch of stamens, a number of them hanging out here. Maybe 6, maybe 8, something along those lines. The stamen is made up of 2 parts. It has a long, skinny part. That long, skinny part is called the filament. I did not label it F because of filament, but lucky choice. And then the round, thick top part is called the anther, and that's where the pollen is. So the pollen is produced on the anther. So again, stamen made of the filament and the anther. All the stamens together create the whorl that is known as the male whorl, and since it is male, we call it the androsium. There we go. I did not write these down, so I have to actually spell them from the top of my head, which is always very difficult for me. All right, now we're getting all the way to the inside whorl. And although whorl sort of seems like something that should go around, in this case, often it's just one large thing in the middle, or sometimes one large thing split into a bunch of parts, which we'll mention, and this is where all the female stuff lives. You'll notice there's a lot more of it in this case. All right, speaking of it being the female stuff, I'm going to start actually with I. I is where the ovary is, and you'll notice that that's very, very far down in the flower. And actually, a lot of times it's all the way down, like it was a little hard to draw this way, but such that it sits inside of where the petals and the sepals are. So the ovary goes way, way, way down. The large structure that is going to contain the ovary or ovaries, because there are a lot of plants that have multiple ovaries, is going to be the pistil. So B here is the pistil. And the pistil is made up of pieces. So if they have multiple ovaries, then typically it's multiple parts that are stuck together next to each other. So whole pieces, we often call those carpels. So a plant could have one or more carpels. And then however many carpels it has, all of them make the pistil. And then finally, we have the parts of the carpels or the pistil. So the ovary was down at the bottom. The long part we call the style. So that's the long part of the female section. And the top we call the stigma. And the stigma is where the pollen sticks to. See, that's a good way to remember it. But also where it can get into and go down the style to reach the ovary. Since these are all the female parts of the plant, the inside final whorl is called the gynosium. So gyne for female, andr for male. And that's how we get all of the crazy parts of a flower. So just in case you didn't know all of these parts, now you do. All right, so let's talk about this process of pollination as it may occur. At some point, we'll also talk about the process of creating pollen and things like that, but we're not going to quite get that complicated right this moment. Right this moment, I want to think mostly about, hey, how do we actually get pollen to an ovary? These are all the possible steps. Our goal is to put them in order. So the first thing we need to do is we actually need to get from regular plant to gamete in some manner. And don't forget the gametophytes in these larger plants, gymnosperms and angiosperms, are much tinier, but they still occur. And so the very first step to reproduction here is meiosis. We have to actually create the things that we need. So here's our step number 1. Meiosis occurs in the mega and microsporangia. So the megasporangia is in the ovary, the microsporangia are on the anthers, and that creates the embryo sac, which is 8 cells in the female section, and pollen grains. And it creates numerous pollen grains, but each pollen grain ends up having 2 important parts by the time it's done. It undergoes meiosis in a manner that creates multiple cells stuck together, and that's important because we're going to have multiple nuclei. All right, so now that we've created the pollen, we've got to do something with it. In this case, the pollen is going to travel via wind or animal, insect, from the anthers to the stigma. It could be from the same flower that has the anthers, but most of the time it travels from one place to another, so we can get a little bit of differentiation. And as we have noted in past times, diversity is very important. All right, so once the pollen gets there, then we need to get it to go down the style. So number 3 is indeed that a pollen tube grows down the style. So the pollen sits on the stigma, and actually it will start to grow a section of it that allow the nuclei that make up the sperm to travel down all the way to the ovary. They really don't take any chances. Things don't just fall. They stop, they know what they're doing, and they get where they're going to go. All right. But as they travel, they're going to make it all the way down to the embryo sac. And in the embryo sac, there is an opening. It's called a micropyle. And we will have a release of 2 little sperm that each came from the same pollen grain through the micropyle. 2 things. I wonder what we're doing with 2 things. Well, it turns out what we're doing is what's called double fertilization. 1 sperm goes and hangs out with the egg, and the egg is obviously the important thing. The sperm and the egg come together, they make a zygote, and it's going to do its thing. The other sperm goes and hangs out with 2 little cells that are called polar nuclei. They're hanging out in the embryo sac. So I said the embryo sac had 8 cells. It has 5 of them that are related to sort of the front and the back ends of the embryo sac, and they go away after this is all over. But the 2 polar nuclei work with a sperm to form the endosperm. Which makes this really interesting is it means that the endosperm is made from 3 tiny cells, each of which has 1 set of genetic code. And the endosperm is what we call triploid. So diploid is a normal plant. Haploid is our gametes, so our sperm is haploid, our egg is haploid. Each of our polar nuclei are a haploid. Endosperm is triploid. Now, it cannot turn into any kind of actual functional plant, but it does create food for the embryo. All right, so we had said, though, that the other fertilization formed a zygote, so clearly the next step, if you have been following along here, is that 6 says the zygote undergoes mitosis, because now it's already diploid, to create an embryo with a radicle, which is a tiny piece of a root, and a cotyledon, which is essentially like its first leaf. But it's also a way to grab the endosperm, which is the food, and get it into the important parts of the embryo. You could have 1 tiny cotyledon leaf, or you could have 2, which is how we get dicots and monocots. We'll talk about those more in a moment. All right, finally, we want to keep this embryo safe, so a seed coat forms around it. It maintains both the embryo, the radicle, the cotyledon, and the endosperm all within the seed. And then the last thing that's not really on here is, of course, that the ovary ripens around the seed. It is released from the plant, and it can travel someplace. And depending on what kind of ripened ovary it is, depends on how it gets somewhere. So it might travel through the wind, like little maple leaf helicopters. Those are essentially fruits. Or it might be something squishy and delicious that somebody eats or an animal eats and then is able to either spit or poop out the seed somewhere. It might be a nut that a little squirrel takes and moves somewhere else. So the seeds go and they do their thing. So that's all of our stuff. Meiosis, creating our pollen. Pollen traveling from anthers to stigma. Pollen tube growing down the style, releasing the sperm through the micropyle, which is in the embryo sac. Double fertilization to create a diploid zygote and a triploid endosperm. And the zygote begins to grow into an embryo, which has a small root and a small leaf, 1 or 2. And then finally, the seed forms around it, and it can go off and become its own plant. Super exciting. All right, but now we really need to make sure we know which parts of these are indeed sporophyte and gametophyte, because this sometimes gets lost in the shuffle since the gametophyte is so small. So we have to think about which of these items were essentially diploid and which parts were essentially haploid. All right. I am going to go ahead and attempt to hunt down all of the ones that are definitely haploid so I can point them out to you. So for example, we already mentioned the polar nuclei. They are some of the cells in the embryo sac. And the embryo sac is 8 definitely haploid cells. They belong to the gametophyte. Interestingly enough, synergid is another set of cells in the same embryo sac. So we've actually talked about now almost all the cells in the embryo sac. The synergids are down at the micropyle, and then there's the antipotals on the other end. Egg cells, really, they contain the most important things since they're going to become the new plant. So they tend to get a lot of helpers. In this case, 7 helpers: synergids, antipotals, and polar nuclei, just to make sure that everything goes as planned. So I mentioned already that pollen is clearly a haploid item. It is haploid, so it belongs over here in gametophyte. And more specifically, inside the pollen, I said there are two sperm, and we give them names. And we call them nuclei because they're nuclei within the pollen. One of them is the tube nuclei. One of them is the generative nuclei. The tube one, I believe, is the one that will attach to the polar nuclei to create the endosperm, which you do see up there, but we're not dealing with yet. All right. So now on the other hand, let's think about the other things we've got. Well, an anther is a part of the major plant. Even though it produces pollen, it does so by meiosis. Therefore, the anther is actually an important key player in the regular sporophyte part of the plant. Same thing goes for the ovule, which holds the ovaries. Even though it seems like the ovule might have some of these haploid things, in reality, it's a key part of the sporophyte. And so it's diploid and goes over here. The seed, as we mentioned, is what happens after fertilization. So if it's a zygote, it's definitely going to be part of the sporophyte. And in fact, it grows into the whole new sporophyte. Within the ovule is the megasporangium. And here's where this gets important, because I only have one of these on here. But if it's a sporangium, it's producing spores by meiosis. If it is a spore, it was produced by meiosis. This is where it gets tough. So if you see anything that ends in sporangium, always sporophyte. If you see anything that ends in spore, always gametophyte. And then finally, what do we do with our weird little endosperm? I mean, in reality, it technically belongs to the sporophyte portion of the plant, but do remember that it's triploid, because it's a weird one. All right, so now you know where all these different things belong, and if you're looking at an image of the reproduction, you should be able to say, oh, sporophyte, gametophyte. Just remember the gametophytes are very tiny. All right, I said we would get back to monocots and dicots, and here we are with the key concept that the difference is that little starting leaf. But that's not the only difference. It is just a part of the differences. Now, the whole thing with these is that they're essentially genetic lineages. So one type of plant went this way and has these characteristics. The other type went this way and has these characteristics. And as far as we can tell, that's mostly true. That dicots especially are like their own little lineage. But we're always learning new things about plant genetics. This could change tomorrow. But the most important thing about them, of course, is that both of these are flowering seed plants in all cases when we talk about monocots or dicots, flowering seed plants. So we're not going to talk about pine trees with flowering seed plants. Pine trees are gymnosperms, neither monocots nor dicots, which is, of course, why we don't look at their leaves for things. So we already mentioned that the key concept here, of course, is that monocot is one cotyledon, and the dicot is 2 cotyledons. But there's a bunch of other things that make them different as well. And as groups of plants, they have different levels of importance to us. So monocots are considered to be these sort of evolutionary predecessors. Think that they came first. They seem to be a little bit more simple. and they have a lot less stuff going on. So I'll discuss them quickly. The main monocot that you are probably most familiar with is grass, interestingly enough, and things in the grass family. Now, this is where it gets a little bit higher and more complicated because things in the grass family include stuff you might not think of as grasses, such as wheats and barleys and things like that. So those are monocots. The other thing in this family is corn. So all of your grains, which in my case is all the things I don't eat anymore. But you do you. Here's the neat thing about it. If you pick up a blade of grass, preferably a thick one, or a corn leaf, what you'll find is that the veins in the leaves are parallel, which is kind of neat. So parallel veins in leaves is a key concept of monocots, along with having just that one starting leaf. Oh, as a note, because of the way seeds work, one cotyledon in a monocot seed means that the seed will also have one piece. So seed is in one piece. If you think about corn, this is a good example. Corn seeds are corn kernels, and it's just one piece. But with two cotyledons, the seeds are often in two pieces. And the best way to think about that tends to be beans. If you think about a bean, it comes in two halves, and those halves can actually break apart. If you're not sure about that, go find yourself a lima bean and have a little bit of fun with it. They do break apart. All right, so parallel veins in the leaves for monocots. Dicots have what we call branched veins, which means that the vein comes down the center and then pieces come off of it. So let me sketch each of these for you because it'll be helpful. So if this is my corn leaf, its veins look a little bit like this. And then if this is my dicot leaf, its veins look like that. All right. This vein differentiation actually extends to the stems and roots as well. So that's kind of neat. So the stems have clustered veins. So they're kind of just all over the place. Or actually scattered is probably the better word. Have scattered veins. In monocots and in dicots, the stems have veins around the outside. The best way to think about this one is that almost all trees that are angiosperms are dicots. And so they have rings. Those rings that are in the trees, that's how that looks. Whereas in monocots, if you cut the stem, you just see like little scattered pieces. In the monocots, the roots have a ring of veins with pith, which is just ground tissue containing typically water or starch in the middle. And dicots, and some of these things will actually become more important later on when we start looking at plant structures. Roots have veins right in the center, often in an X shape. So in this case, the veins are smack in the middle. So things to keep an eye out for. And then the final thing that you might notice between monocots and dicots is that leaves are often in multiples of 3. Actually, leaves petals. And in the case of dicots, leaves and petals often in multiples of 5 or 7 or something else, but not 3. I know, they're all odd. It's super weird. But those are some of the ways you will tell your monocots from your dicots. And we'll get back to those again when we do plant structures next time. All right, the last couple things we're going to think about is how plants interact with us and the world. Now, plants, as we've mentioned before, can't move. That is the key thing that makes them different from animals. Since they can't move, they need ways to defend themselves. And often, because we are coming by and doing things with them, they're real big on, defending themselves. So what are some things plants do to defend themselves? Well, think about plants you've encountered. For example, there are bushes in my neighborhood with spines. Spines are a way they do that. As well as any tough outer coverings. They might have various kinds of other prickly things like barbs, they might produce nasty tasting chemicals. They especially do that in some cases when you actually go ahead and cut them. Sometimes if you cut plants, they produce nasty tasting chemicals. They also produce, in some cases, toxic chemicals. So they don't want to be eaten, but they also will produce things that are trouble. So we talk about phytochemicals as the chemicals that come from plants. And some of them are phytotoxins. So they're plants you can't eat because they produce things that will actually stop you. There are some that seem to be beneficial to humans, but not all humans. So, like, for example, some humans do well with certain phytochemicals and some do not. And in some cases, those phytochemicals are better if we, for example, spend a little time breaking them down. So lignin is a part of many plants, and it's a very tough tissue for humans to break down. But, for example, if we cook things for a long time, the lignin is easier to use, and then it's less troublesome. And of course, humans have been co-evolving with plants for many, many years, and we've co-evolved differently depending on where your ancestry is from, which plants you grew up with, that kind of stuff. So these are just some plant defenses. But of course, not all plants are bad. Let's end this by thinking about the things we actually do with plants that are beneficial. Some of them are beneficial to us and not to the plants. So for example, we use them for building, we use them to make paper, we use them for many other types of supplies. We just have to be careful not to overuse them. We also use them for medicine. It turns out that some of those chemicals are made to, for example, destroy other things like microbes, and we can use those. Or they actually work well within a human system to cut back certain chemicals that cause us inflammation. We also enjoy plants. So plants are good for decoration. They're good for revitalization. Nothing feels better, honestly, than getting out in bare feet and running around on plants. They're real good stuff. So there are plenty of benefits. We don't want to get rid of all of them. Oh, and of course, the big one is that they produce oxygen. We still need that. That's a key thing. We can get it from cyanobacteria too, but I really like the plants. So even though they might be trouble sometimes, they are also beneficial. And I hope you learned a little bit about them here. So enjoy.